Hello, everyone. I'm Yilei, a postdoc in Sedlazeck Lab at Baylor College of Medicine, Human Genome Sequencing Center. First, I want to thank the labs for giving me this opportunity to present this as precision medicine event. And today, I'm going to talk about our work with computational analysis of DNA methylation from long-read sequencing. The DNA methylation on human genome refers to methyl groups being added to DNA molecule. The figure on the right illustrates that the methylation of DNA could cause nucleosomes to pack tightly together and make the transcription factors cannot bind the DNA, which leads to genes being not expressed. This is a simplified basic function explanation of how DNA methylation regulates gene expression. And those gene expression alterations could cause, for example, the repression of transposable elements or the silencing of tumor suppressor genes, which could lead to cancer development. DNA methylation is also related to genomic imprinting, X chromosome inactivation, and aging. So in human, DNA methylation exists mostly in the form of cytoside methylation that happens on CpG locations on the genome, which usually we call 5-MC mostly. And there are three ways to sequence DNA methylation in the human genome: microarray, short-read whole genome sequencing, and long-read whole genome sequencing. Here in the figure, it shows the short-read whole genome sequencing and microarray usually requires a base conversion step with treatment that converts unmethylated C to U. After sequencing, microarray could profile at most 935,000 CpGs on the genome, while short-read whole genome sequencing could profile the genome and DNA methylation, where there are around 28 million CpGs. The conversion process itself and the converted results make the entire process hard and complex. On the other hand, long-read sequencing could directly call the DNA methylation during the base calling process, which means it provides the simultaneous readout of both nucleotide bases and methylation. In the meantime, long-read sequencing technology obtained other benefits, such as genome phasing and structural variations calling. So on the table, we can see that the length improvements and other advantage from third generation sequencing, which is the long-read sequencing here. So there are two platforms that we are currently using for long-read sequencing. PacBio and Oxford Nanopore Technologies can both provide native DNA methylation readout. They take similar approaches which use the deep learning method to profile the different signal behavior between modified bases and canonical base. So more specifically on the left, PacBio learns the pattern of unique pulse widths and interpulse durations for different kinds of DNA modifications. While ONT on the right identifies electrolytic current changes for the modified bases. Until now, PacBio achieves a better accuracy with detecting 6MAs, which is another type of DNA methylation that does not exist in human. And ONT achieves a better accuracy for 5MCs on human genome. With this direct detection ability, no additional steps are needed during the base calling stage, and we can access DNA methylation right after it. Again, only long-read sequencing here could provide the simultaneous readout of nucleotide bases and DNA methylation. With this knowledge, we further looked more into how to perform DNA methylation analysis on long-read sequencing data. Here we have recently published a review paper about it at Nature Review Genetics, where it summarized the major steps and applications for the computational analysis. So here we start with the major steps of DNA methylation analysis. The input sample usually consists of cell lines, tissues, population samples, or cell-free DNA. Before long-read sequencing, an optional treatment step can be applied to mark chromatin structure or cell-free DNA extraction, for example. Then we start with library preparation, sequencing, and base calling step. The base calling step could be performed with the sequencing company software, but there are other third-party software that could prefer more DNA modification types. And after sequencing, reads are aligned to the reference genome to identify or base call methylation modifications across the genome. Long reads enable efficient variant calling that includes single nucleotide polymorphisms, which we call SNPs, insertions of deletions, tandem repeats, and structural variations, further leading to optional genome phasing and methylation signal smoothing steps. For comparative methylation analysis, differential methylated regions detections is usually performed to identify allele-specific methylation, and tissue type and cell type differential methylated regions. Cell type deconvolution and identification of population signatures can also be achieved with computational methods. So those results further leads to various post analysis. So the colors in the boxes represents which type of input samples use the steps and generates the results in post analysis. Then we want to dive into the applications of long-read methylation analysis. So genome-wide methylation signals detected and compared across samples of haplotypes provides several insights. First, it could be used for identifying differential methylated regions between normal and tumor tissues. And here we have reviewed the hypermethylation of a promoter of a tumor suppressor genes, which leads to transcriptional silencing. Additionally, long reads can enable differential methylation analysis of complex regions such as tandem repeat in the FMR1 region. Let's list an example here with high and low methylation, red and blue respectively. And also, it enables to detect allele-specific methylation on two long-read phased haplotypes can inform regular mechanisms such as gene printing between males and females, which entails hypermethylation and one promoter copy that affects gene expression. So allele-specific methylation can also improve genome scale methylation quantitative treated loci studies. And also, cell type deconvolution from a mixed DNA methylation signal across the tissues reveals the cell type composition and can inform DNA methylation atlases. And also, long-read methylation data can provide other genomic insights, such as cell-free DNA that enables rapid classification and tumor cell abundance estimation in blood samples. And additionally, marking DNA in open chromatin methyltransferase enables the detection of chromatin structures by examining message status. So to go into more detail part, here we show how exactly the access of DNA methylation on long reads is usually performed with read alignment, where the reads are aligned to the reference genome. A score on each CpG location on each read is reported, which can be visualized like that, like what I'm showing in the slides. So the red dots here means the CpG is methylated on the reads. Well, when there's no color, it means the base in the location is not methylated. With this convenient reporting and visualizing standard, we can directly utilize the DNA messaging signal, just like how we treat other signals that we get from read alignment, such as SNPs, SVs. Here, we have seen many papers even before the fast development on long-read sequencing technologies that use the measurement of DNA methylation to, for example, correlate with transcriptions, disease, aging, or lifestyle differences. However, these tests could be performed even without long-read sequencing technologies, which raises our first question: how could we actually use DNA methylation signal that we read from long-read sequencing other than just measuring? And a unique signature of DNA methylation is it is different among cell types in some regions. And the left figure is a simple illustration of immune cell types. And the DNA methylation landscape are different. So this brings us another question to solve. So given a more complex bulk sequence tissue sample, can we actually use DNA methylation signal to identify cell type specific mutations? In here, we gather long-read bulk sequence tissues, check DNA methylation differences, and try to associate those with mosaic structural variations to prove that the mosaic variants in the box sequence sample are indeed cell-level signals. A recent publication about DNA methylation landscape in cell types reveals the atlas of a map of differential methylation regions around among cell types. We call those regions cell type specific DNA methylation. Based on these, we further try to associate the mosaic variants to cell type specific methylation. So we talked about a bunch of mosaic variants, and what are these? Mosaic variants are those that occur after fertilization and we are not and are not inherited from our biological parents. Somatic changes occur for many different reasons and can grow as cell grows and divide. With our lab's previous method, SniffCell, we showcased the capability for detecting mosaic structural variations in bulk sequence human genome. In this figure, we are seeing with long-read bulk sequence data, we are detecting mosaic insertions and deletions in tissues. However, we found those mosaic SVs, but without any cell type information. So the interpretations of those mosaic variants are actually limited. Then we naturally just wonder if we can annotate those mosaic variants directly from bulk sequence tissues. So our first move is actually to combine our lab's previous work, SniffCell, mosaic as we call it, from long-read bulk tissues with DNA methylation analysis, and we call the tool SniffCell. So in here, we with a benchmarking cell line mix sample, we can effectively locate SV supporting reads with DNA messaging changes. So in here, the blue boxes we kept are a mosaic insertion detected by SniffCell, where we see a strong DNA messaging change around this mosaic insertion. However, the Human mix is just a cell line which we know the messaging of those are different from tissues. Then we further wanted to see if we can see a mosaic SV in real tissue that associate with DNA messaging changes. We have located several examples where one of those located on a gene that's related to neurogenesis. So SniffCell have identified a mosaic deletion with 10% allele frequency in the cerebellum tissue, and the supporting reads of this mosaic deletion is showing a universal low DNA methylation comparing to the ones that have high DNA methylation. We have seen the effectiveness of our program in tissue samples. Then we want to know how accurate it could be. So to check our program's accuracy for identifying DNA messaging changes around mosaic SVs, we circled back to our cell line mix sample for benchmarking. So we compared our somatic SV coding against the germline SV coding in HPLC cell lines, along with the DNA message measurements. This is a tricky thing to do because in the Human mix, most of the mosaic SVs are only supported by around 5 to 7 reads. And in the mix, and we're actually comparing those with rest of the reads, which are usually more than 90 reads. But even with this, we are still achieve the decent false positive reads. The false positive here means our identified mosaic as we correlate with the methylation changes in Human mix are not supported by HPLC cell lines. But is this all we can do with long-read DNA methylation? So with single-cell RNA technology, we could obtain the cell types from a tissue and further look up their DNA methylation landscape. So as I mentioned before, a recent paper has released a DNA methylation atlas for normal human cell types, where it contains the regions on the genome where a certain cell type have lower methylation level, while the others have higher methylation levels. In fact, we have already identified regions across the genome that shows differential methylation pattern in major cell types in brain. For example, in here we have found several locations where glial cells and neuron cells are showing differential methylation patterns. On the right, we see in those locations, glial cells are usually having super low methylation compared to the neuron in those regions. So if we take the first location a closer look, when we are matching the identified region from the atlas to our smart brain sample, we could assign the reads to a cell type based on its methylation signature. From single-cell RNA-seq and DNA methylation atlas that we just showed, we showed the reads with higher methylation levels could come from neurons, while the lower messaging levels could come from glial cells. So furthermore, to rule out the possibility of this, this is actually a haplotype-specific messaging. We have already faced the recent haplotypes, and we still see the messaging differences inside each haplotype, which means those differences are not haplotype-specific messages, but are real cell type differences. So based on that finding, we actually utilize EM algorithm to assign the reads to cell types. We here show that we can separate the reads based on their DNA message signature in the given regions. With regions that have a larger DNA message entropy among the cell types, the EM algorithm can decode more cell types other than just these two. So till now, we talked so much about methylation-based read classification and cell type assignment, but how do genomic variants look like in those reads from each cell type? So take this plot as an example. We see the reads amount of neurons glial cells almost 50 to 50%. So if you are only counting the allele frequency of a variant, how should we know that this is a heterozygous variant or a cell type specific variant? Here we removed all the DNA message color in the IGV plot, and the blue dot here is a mismatch in the read length, which could further be considered into SNPs. So on the top panel, we see the SNPs is actually a heterozygous variant, and with our message-based read clustering, evenly distributed in different cell types. We are assigning the reads into 4 different cell types in a different region with more cell type level DNA message and differences listed in Atlas. So based on this, we if we take a step forward, the question is, can we actually find an somatic variant based on DNA message and classifications? So to explore the potential of that, we ran it on the deep ONT sequenced brain with 2 regions. We obtained 2 samples, one from brain frontal cortex, another one from brain cerebellum. And to answer the previous question, that if we can decode DNA somatic cases, the answer is yes. We here are showing the potential of our approach that capable of identifying the somatic SNVs in 2 deep sequenced human brain. So these are 2 ONT deep sequenced brain tissues from the same individual. The top IGV screenshot is from cerebellum and the bottom one is from cortex. With our approach, the first thing that we see here is that there is a huge cell type proportion differences in those 2 regions, whereas consistent with single cell RNA analysis and the common knowledge of the cell type proportions of those tissues. So furthermore, because of these 2 samples, we are seeing from the same individual, the somatic SNVs observed in neuron cells in the cerebellum should also be observed in cortex. So the blue dots here, we are still seeing mismatches in read elements where we can be consolidated into SNVs. So here we can see that the mismatches only exist in our classified reads from neuron cells, which means the same cell type in 2 tissues in the same individual contains the same somatic variants. I think the most important thing is, with the current allele frequency-based somatic variant detection method from bulk sequencing data, we are almost certain the somatic SNV we saw in the top panel tissue will be identified as germinal variants, which they are actually in fact somatic mutations. And with some deeper observations, we see those somatic SNVs in cortex neurons happens with less frequency, which still makes sense because the neuron subtype and other mixed cell types in the cortex are actually more diverse than cerebellum. And as we show in the last 2 slides, in some regions where different cell types show in the distinct methylation landscape, we could classify more cell types. And based on that, we can further systematically assign variant cell type based on the allele frequency match. Here we obviously have a bunch of filters to make sure we don't call false positives too much. And then we went into assign mosaic as with cell type according to its closest block, DNA messaging profile. With that capability, we are able to identify genome-wide mosaic as with cell type based on DNA methylation. Here we show an example where the mosaic deletion is correlating with oligodendrocyte proportion, where it suggests this mosaic SV could be a cell type specific one. To summarize today's presentation, so we first introduced an overview of computational analysis of DNA messaging from long-read sequencing platform. We discussed long-read benefits such as direct message encoding and general steps for DNA message analysis on read data. We then discussed about several applications where we can utilize the DNA message signal. We then gave a detailed example from our lab's work about the application called SniffCell, where we use a cell level analysis method for mosaic variance cell type identification based on DNA methylation. So finally, I would like to thank the opportunity again that LabRoots gave me to present our work at this meeting. I want to also thank the members in Sedlazeck Lab and the production team in our center. The project is supported by NIH Smart Consortium and CAR initiative. We also perform cell sorting process that is performed by UCL and being sequenced by Oxford Nanotechnologies. With that, thank you all for listening, and I'm happy to take any questions through the LabRoots.